Hey guys, this is Mr. Wayt, and today I want to go over the cell communication and cell cycle study guide with you. And this is normally what I would do the day before class, but it can't be there. I apologize. If you do have any questions, be sure to email me and we'll see what we can do. Let's start out with cell communication. Cells communicate with one another through direct contact. We want to make sure that we understand that is cell to cell. They're actually touching each other. In fact, I would connect these two lines right here. Cell to cell contact is this direct contact. We talked about that yesterday when we have antigen presenting cells in the immune system. They present a piece of the antigen to another cell, and that presents it to other cells. That's how you develop antibodies. That's how you get killer T cells. But that's a great example of direct contact. The other thing we see is that cells communicate over short distances, and this is the key word, by using local regulators. These are very specific. They're for one cell to another. An example of this yesterday was the neurons in the nervous system, how even though they're not touching, there's a synapse between those neurons, and they can go through that space. It's a very specific signal for a very specific cell that's in the localized region. The last thing over here, some can travel long distances. And when you think of long distances, I hope you think of hormones. The one that we looked at yesterday specifically was the human growth hormone, how that's released from the brain. It goes through all parts of the body. Even though it has a specific receptor for a specific cell, it's released in the blood. It goes everywhere. So that's a good example of that. Guys, I apologize. I'm learning how to use this technology. Black may not be the best color for this because it looks like I have a floating head right now. But let's keep going with this. Introduction to signal transduction pathways. These guys link signal reception, and that's our first word right there that we need to make sure we understand is reception. And that's going to elicit a response. Many signal transduction pathways include protein modification and phosphorylation cascades. Remember, phosphorylize something means to either add or take away a phosphate, turning it on, turning it off. And we could modify a protein, or we could also be going straight to the DNA and causing a major change in gene expression. It says the signal begins with reception. I think we already identified that. Okay, just make sure we know reception of a chemical messenger, usually in the form of a ligand. By reception protein to the target cell, the ligand bind domain of the receptor. It recognizes this. Remember, it has to be like a hand and glove fit, a lock and key, which can be a peptide, small chemical, protein, specific one-to-one relationship. I think that's what that's getting there. It's very specific. An example of this would be a G protein coupled receptor, and looks like there's some formatting issues here. Receptor protein in eukaryotes. Signal cascades are signals from receptors to cell targets, often amplifying. Make sure we understand that these signals can be amplified. So they start out small and get large, resulting in the appropriate response. So there's another word there. We didn't necessarily go in order this time, but ultimately we are eliciting a response, which could induce cell growth, include cell growth, secrete molecules, or gene expression. After the ligand binds, intercellular domains of a receptor protein changes. So guys, don't forget, as soon as this receptor meets with a ligand, we're going to get it to change shape. That's the receptor might be changing shape. They often call that a conformational change. That's going to start this transduction of the signal, which is our third word down here. So we have reception. Let's identify this. Reception. And then I don't want you to forget this one. Transduction is number 2. Let's go ahead and circle that one right here. And then that leads to some type of a response over here. Did we finish anything here? Oh, we can't forget there are these very important second messengers. The most common one is cyclic AMP, often abbreviated CAMP. They're molecules that relay and amplify the signals. Let's see if we've got anything else here. I feel like that just got dark on me. Let's bring that back up. Actually, I need to go up here and lighten that up. Okay. Transduction pathway influences how it responds to its environment. That's what it's all about, the internal cell responding to environment. Signal transduction may result in changes in gene expression. Now notice if it did that. If we're going to result in gene expression, we had to get to the DNA, and if you're getting all the way to the DNA, you are going to have some type of a major response, much larger than just altering the shape of a protein, which may alter the phenotype, program cell death, and this is always a good word to know, especially on this upcoming test, apoptosis, programmed cell death. 4.4, changes in signal transduction pathway. They're often elicit a response, which we talked about here. I guess this would be where I should circle number 3, okay? The response down here. So we said again, reception, transduction, response for this one. Mutations in any part of the receptor or the protein will affect the downstream parts. Remember we talked about arrows, how, oh, this one could affect this one could affect this one. If there's a mutation right here, that could stop the next part of this transduction pathway. Downstream components, chemicals interfere with any component, may activate or inhibit the pathway, and make sure you understand what it looks like to activate or inhibit with those arrows. Plenty of diagrams on that. So let's erase this one, and let's get to page 2 of the study guide. Looks like this. Let me shrink out of this, go over here, and enlarge that one more time. So page 2, all right, there's the big important ones. I can't draw on those figures, but you can see reception, transduction, response, huge for this test. An example for that is the G protein coupled receptor. That's what we have on the right over there. It starts with a G protein coupled receptor. It's going to have a change in shape when that messenger arrives to that, or that signal, that ligand. That's going to activate the G protein. It's going to travel along the cell into, it looks like, adenylyl cyclase. Ultimately, that's going to yield, after it does all this transduction, a response. And a typical one that we look at for that is the release of epinephrine. So if you want to look up an example, that might be a good one. Let's scoot this up. Organisms use feedback mechanisms. All right, that's another important part of this test I don't think was covered in the sketch notes that we did yesterday. To maintain the internal environments, keep in mind, basically internal environment is maintaining homeostasis. Homeostasis is we want to keep things the same. If it goes up, we bring it back down. If it goes down, we bring it back up. It says if the system is perturbed, if it's bothered, negative feedback, and I'm just going to highlight this over here, mechanism returns the system back to its set target point. And remember, I always like to think of this. Negative feedback, more gives you less. Okay, it returns it back to a set point. These processes operate at molecular level. Positive feedback, however, and that's what we have right over here. Positive feedback. Come all the way back over here. They amplify a response. So if I'm talking about a positive feedback, more is going to give me more. An example of that, two common examples of that are childbirth, or even mammals suckling, drinking milk, producing the production of milk in the mammary glands. Those are both positive feedback examples. Negative feedback, there's a lot of different examples there, but some common ones would be blood sugar levels. If it gets too high, we try to bring it back down. Getting too cold, we try to heat the body back up, but it's about bringing it back to that set point. So make sure you know a little bit about negative feedback. Let's look at 4.6, cell cycle. We've talked a lot about these. Cells divide. That's just a fancy name. The cells, when we're talking about cells dividing, we're talking about mitosis. Transmit genetic information, and again, if we're dividing, we are making copies, daughter cells of this original parent cell. It's an exact copy. The cell cycle is highly regulated. For the growth and reproduction of cells, cell cycle consists of stages of interphase, G1, S, and G2. Remember, those are all interphase. Then we have mitosis and cytokinesis. A cell can go into G0 where it no longer divides, but it can re-enter the cell cycle in response to any cues. Non-dividing cells may exit the cell cycle or be held at a particular stage in the cell cycle. Mitosis, down here, good word to know, is a process to make sure the transfer of complete genome is all the genetic material from a parent cell, gets copied to two genetically identical daughter cells. Mitosis plays a role in, here's our words right here, okay? Growth, repair, or asexual reproduction, GRR. Mitosis alternates with interphase in the cell cycle. It's sequential. It's made of prophase, metaphase, anaphase, telophase. You're not supposed to have to identify those stages necessarily, but you should know what the chromosomes are doing throughout that process. All right, I think we have one more page. Let's look at the third and final page here. And again, looks like some more diagrams with a little bit of stuff underneath there. And it's talking about regulation of the cell cycle. I think we've looked at this image quite a few times. It does say a number of internal controls or checkpoints regulate through the cycle. You remember, you should know the checkpoints are G1, G2, and M, or where we have those checkpoints and what they're looking for at each one of those stages. Interactions between cyclins and cyclin-dependent kinases. Now remember, cyclin-dependent kinases, CDKs, and these are always going to remain even Steven. It's the cyclins that go up and down, that control it from one cycle to the other. When these cyclins bind the CDK, it becomes a complex therapy. That's going to activate us and allow it to go to the next stage. It says disruptions to the cell cycle can result in cancer, kind of a common answer here, or programmed cell death, aka apoptosis. So, guys, I think that's everything you need to know. Again, if you have questions, feel free to email me. Study up on your material, and good luck on the test that's the next class period.